Welcome back to the online TA review. In this video, we'll be reviewing a few methods that are related to this week's material. The topics will be gels, Sanger sequencing, southern and northern blots, which will lead us into talking about exercise A. So first off, gels. In this class, we focus primarily on two types of gel. Now, when we say the word gel, we're usually referring to gel electrophoresis. Don't get this confused with gel chromatography. Really, the only time bigger things travel faster are with gel chromatography. Otherwise, when we talk about gels, think bands traveling through the gel at different speeds, with smaller things going faster. Now, the two types of gel we focus on are agarose gel and polyacrylamide gel. They differ on what they can run on and the specificity. Agarose gels usually is for DNA and RNA. It has the capacity to handle large segments, but loses specificity for doing so. Polyacrylamide gel, which is the PAG of page, usually runs on small DNA strands and proteins. Because it works with smaller segments of DNA, it also gives us specificity down to 1 base pair differences. Knowing that, let's talk about Sanger sequencing, which uses page to determine the sequence of a segment of DNA. Traditionally, this process required one type of ddNTP and the 4 types of dNTP. Because ddNTP are mixing the 3' oxygen, they cannot have additional nucleotides added to them. When ddNTP are used in replication, which we'll talk about more in week 5's material, they produce segments that are prematurely shorter, essentially stunting the elongation of the new strand. We will eventually have many segments of DNA, all of different lengths. The ones that incorporated a ddNTP will be shorter at different places depending on when the ddNTP was used. Because the ddNTP are radioactive, when we run it on a gel, we'll see a band show up. Traditionally, this was done 4 times, each using a different type of ddNTP, one each for A, T, C, and G. If that doesn't make any sense, click on the link I've put in the description below. Because page gives us 1 base pair differences, we'll be able to know the sequence by... Actually, no, you tell me. How could you determine the sequence by one of these gels? What goes fastest on a gel again? And would an early stunt of elongation make a small strand or a big strand?
Okay, now our blots. There are 3 types of blots, southern, northern, and western. Because each are run on different things, they give us different conclusions. I've provided hashtags that I want you to associate these words with. Southern blot runs on DNA and tells you about gene presence. Northern blots run on RNA and tell you about gene expression. If a band shows up in a southern blot, you know the gene is present. If it shows up in a northern blot, it's also expressed. The steps for a southern blot are as follows. We begin by digesting the DNA with a restriction enzyme, which we then run on an agarose gel that usually has ethidium bromide in it, which dyes the DNA as it runs through. After, we denature the DNA and transfer it onto nitrocellulose, which will secure the bands at the location they travel to. By baking the DNA with UV or heat, the DNA sticks, allowing us to add a probe without the DNA being washed away. The probe is a small segment of DNA specific to the gene of interest, which will bind to the DNA if the gene is present. Finally, through audio radiography, we can see which bands of DNA actually had the gene present. The steps for a northern blot are pretty much the same, except we don't have to worry about denaturing the RNA because it's already single-stranded. In addition to adding the probe for the gene of interest, we also have to add a probe for a housekeeping gene, a gene that is expressed in equal amounts in every cell, thus acting as our control. Darker bands mean more mRNA was being made, which usually means that the gene is being expressed more in that area. Okay, now having talked about all of these things, we're finally ready to look at exercise A. Take a second to read through, identify the 4 steps, also see if you can answer the questions that go along with this exercise. Go ahead and pause the video. From the data we collect from the southern blot, the fibroin gene is present in both PSG and MSG. Different sized bands are produced because we used different restriction enzymes, which both make fragments containing the gene but of different sizes. This is because they cut at different places. Because we didn't run a northern blot, we cannot make any conclusions about the gene expression.